Hello, my name is Kalia Miller, and today I will be talking to you about how nature, nurture, and epigenetics influence the development of gender identity. Gender identity is important for any individual to live a happy and healthy life. Gender identity is defined as a person's inherent sense of being male, female, or an alternative gender that may not correspond to a person's assigned sex at birth. Transgender, cisgender, gender dysphoria, and gender incongruence are all examples of an alternative gender. Past studies have shown that gender identity has been influenced by nature, nurture, and epigenetics in different ways. Gender identity develops through a dialectic process of personal reflection and with input from the social environment. Gender stability is referred to as the stage in development where children recognize that gender remains the same over time. Gender stability has been measured by showing children a variety of stimuli and asking questions about gender of each of the stimuli to assess whether gender remains stable over time. Gender incongruence is a condition defined as a pronounced, persistent incongruence between an individual's experience of gender and the assigned sex gender. Gender dysphoria occurs when an individual's gender identity and the assigned sex at birth do not align. During the last few decades, an increase in gender dysphoria has been reported. With gender dysphoria have a higher risk of depression, anxiety disorders, suicide, less access to healthcare facilities, and those who suffer from gender dysphoria or gender incongruence may seek gender affirming hormone therapy to help. Feminization or masculinization may be sought through um, gender affirming hormone therapy. Testosterone is used for seeking masculinization, and estrogen combined with anti-androgens are used for those seeking feminization. Collaborative treatment programs seek to integrate psychological support for youth and their families. These treatments give attention to youth's social, familial, and cultural context. In some cases, hormones suppress puberty or achieve gender expression to match a child's gender identity will be administered. Nature versus nurture debate has been focused on gender identity in many previous studies. Nature is described as the biological and genetic inheritance that influences development or one's gender identity. Nurturing is referred to as social experiences one may have that influence development or one's gender identity. I've included two little pictures there that kind of give you a little bit more of an example of what nature and nurture is. And so Bailey and colleagues examined the extent to which homosexual men's variation in gender nonconformity runs in families by examining pairs of genetic brothers who were both homosexual. Um, they found family resemblance for homosexual brothers in the degree of gender nonconformity. Bailey and colleagues suggest that genetic factors play a role in gender nonconformity variation among homosexual men. Fisher and colleagues found organizational effects influence sexual orientation. Organizational effects during prenatal and neonatal development and are permanent. Fisher and colleagues found evidence of organizational effects and differences in sexual orientation in three different brain regions. The suprachiasmatic nucleus, the anterior commissure, and the anterior hypothalamus. The suprachiasmatic nucleus is larger than the homosexual male compared to the heterosexual male. The anterior commissure is smaller in homosexual males compared to heterosexual males, and the anterior hypothalamus is smaller in homosexual males compared to heterosexual males, and smaller in size compared to heterosexual females. Differences in brain regions show evidence of biological and genetic influences of nature on gender identity. Ristery and colleagues aim to summarize studies on biological basis of sexual development. They focused on the impact of sex hormones and genetic background on the development of sexual differentiation and gender identity. Evidence suggests that sexual dimorphic brain could be the anatomical substrate of psychosexual development, on which gonadal hormones may have a shaping role during prenatal and pubertal periods. They found that prenatal and pubertal sex hormones have a permanent effect on human behavior. On the nurturing side, there is gender labeling, gender schemas, gender categorization, recognition of genitalia, family, familial factors, and peers influences. Children are known to engage in gender labeling, which leads to self-labeling. This can be seen as a nurturing influence on gender identity. Gender labeling tasks have been used to assess whether children are able to discriminate between gender categories. Most children develop the ability to label others according to their gender and to use gender labeling regularly in their speech between ages 18 and 24 months of age. Diamond suggests that a sense of self and actively seeking information begins as well at this age. Children's Children's daily experience is marked by pervasive gender categorization, such as names, clothes, hairstyles, toys, teams, and restaurants for boys and girls separately. The development of gender schemas or stereotypes relies on socialization, where children learn what it means to be female and male within a particular cultural context in their respective environments. Um, Federici and colleagues found female attribution when the vulva appeared in the human picture half of the time compared to male attribution when the human body showed the penis. They also found that female cue is recognized only in the absence of male cues. They concluded that cultural stereotypes and prejudice affecting gender attribution to be a cultural product and consequence of evolved cognitive biases. They also suggest that genitalia and face are what guides an individual in gender attribution. Some other researchers examined the prevalence of gender dysphoria among twins and non-twin siblings of individuals with gender dysphoria. Familial factors are confined to shared environmental influences during the uterine periods. Food preferences, traditions, language are all considered to be familial factors. All gender diverse children, adolescents, and adults face challenges related to social and family acceptance. Some other researchers examined peer influence on the four aspects of adolescent gender identity in racially and ethnically diverse 7th and 8th graders. They found that significant peer influence on between gender components of gender identity intergroup bias among 7th graders and 8th graders. They also found that students felt pressure for gender conformity among 8th graders. Adolescents who were more content with their gender had a higher number of friendships than those who held more negative attitudes than other genders. Epigenetics is the study of how long-lasting gene-regulating attachments are placed and removed. Carla and colleagues carried out a global DNA methylation analysis comparing gender incongruence of transgender people before gender-affirming hormone therapy to cisgender people. They found that cisgender people and transgender population have a different global cytosine phosphate guanine methylene prior to gender-affirming hormone therapy. They suggest that epigenetics may be involved in the etiology of gender incongruence. There are multiple genes, I have listed them there, that could be involved in brain development and epigenetic factors that play a role in differential development that might be related to gender incongruence. Gender incongruence is a complex multifactorial trait involving intricate interactions between sex steroids receptors, genetics, and epigenetics. Shepard and colleagues profiled genome-wide DNA methylation in blood of transgender women and transgender men before um, uh, gender-affirming hormone therapy. Their study was the first epigenome-wide analysis of transgender and transgender women during gender-affirming hormone therapy. They found that X chromosomes and specific autosomal DNA methylation patterns established in early development are refractory to change in association to gender-affirming hormone therapy. There is evidence that gender-affirming hormone therapy induces unique blood methylation signatures in transgender people, feminizing and masculinizing gender-affirming hormone therapy alters the blood methylome in progressive manner. Sex-specific DNA methylation is present throughout life, determined by genetic and developmental programs, and are not susceptible to change in responses to hormones. In conclusion, previous studies and examples have shown nature, nurture, and epigenetics have a potential influence to the development of gender identity. Gender labeling, gender schemas, gender categorization, recognition of genitalia, familial factors, and peers have a nurturing influence. Gender nonconformity in homosexual men, organizational effects, and anatomical brain differences are known to have a genetic influence. There is epigenetic evidence that specific genes may be involved in gender identity or incongruence, and there is also epigenetic evidence that DNA methylation of blood after GHT induces a unique blood methylation. It is important to acknowledge all of the influences on gender identity because gender diverse individuals suffer from clinical and social changes. Thank you.